**** Microbes in your gut rewrite DNA? No. They don't just digest food. ***, they tweak the marks that silence genes. How do tiny bacterial molecules hijack a human enzyme? The answer is a chemical battle inside every cell. And the stakes? Your gene's future. DNA methyltransferases add a methyl group to cytosine bases. The enzyme grabs S-adenosyl methionine SAM and flips the target base out of the helix. A cysteine residue then steals the methyl, forming a covalent bond. Once transferred, SAM becomes homocysteine and the DNA returns to its shape. Microbial metabolites can slip into this pocket or tug on distant hinges, altering the enzyme's grip. Short-chain fatty acids, indole derivatives, and polyketides all compete with SAM or shift the protein's conformation, reducing methylation rates. Pause 0.8 DNA methyltransferases transfer a methyl group from S-adenosyl methionine to cytosine. The enzyme first binds SAM in its active site, positioning the sulfonium carbon for attack. A conserved cysteine residue acts as a nucleophile, forming a covalent intermediate with the methyl group. Simultaneously, the target cytosine flips out of the DNA helix into the enzyme pocket. The base is reoriented so that its N3 atom attacks the methyl carbon, creating a transient methylated intermediate. A conserved glutamate stabilizes the transition state and facilitates proton abstraction from cytosine. After methyl transfer, the covalent bond between cysteine and SAM breaks, releasing S-adenosyl homocysteine. The enzyme then reverts to its original conformation, allowing the DNA strand to return to its helical structure. This stepwise process ensures precise placement of a methyl group at CpG sites across the genome. Understanding these mechanistic details is essential for interpreting epigenetic regulation in health and disease. Pause 0.8 DNA methyltransferases use S-adenosyl methionine, or SAM, as the methyl donor. The catalytic pocket binds SAM tightly through hydrogen bonds and hydrophobic contacts. Certain microbial metabolites, such as butyrate, can enter this pocket by structural mimicry. Butyrate lacks a sulfur atom, but fits the shape of SAM's adenine moiety. Another metabolite, 5-methylthioadenosine, resembles SAM's ribose and methyl group closely. When these metabolites occupy the pocket, they block access for true SAM molecules. This direct competitive inhibition reduces the rate of cytosine methylation in DNA. Kinetic studies show a dose-dependent increase in IC50 values as metabolite concentration rises. Graphical overlays illustrate how butyrate and 5-methylthioadenosine align with SAM within the active site. Thus, microbial metabolites can fine-tune epigenetic regulation by physically obstructing methyl transfer. Pause 0.8 first. We view the DNMT in its unbound or APO state. Notice the flexible loop that normally positions the catalytic pocket. Next, introduce a small metabolite at a distal allosteric site. The molecule does not touch the active center directly. It instead pulls on a hinge region connecting two domains. This movement tilts the catalytic domain away from DNA. As a result, substrate access is hindered and reaction speed drops. Thus, the enzyme's efficiency is lowered without blocking the active site. By comparing these two states, we see the power of conformational control. Such allosteric modulation illustrates how metabolites can fine-tune epigenetic activity. Pause 0.8 Gut microbes produce short-chain fatty acids that can enter host cells. These metabolites activate peroxisome proliferator-activated receptors, which upregulate folate transporters. Enhanced folate uptake increases tetrahydrofolate pools used in 1-carbon transfers. Tetrahydrofolate donates methyl groups to homocysteine, forming S-adenosyl methionine SAM. Higher SAM concentrations provide more substrate for DNA methyltransferases. Conversely, microbial depletion of folate reduces SAM synthesis and lowers methylation capacity. This shift is visualized in a metabolic network diagram linking gut activity to host 1-carbon flux. Bar graphs show SAM levels rising when specific metabolites are present versus absent. Thus, microbial metabolites indirectly modulate DNA methyltransferase activity through donor availability. Understanding this pathway helps explain epigenetic changes linked to diet and microbiome composition. These insights underscore the importance of maintaining microbial diversity for epigenetic health. Pause 0.8's DNA methyltransferases add a methyl group to cytosines and CpG dinucleotides. When microbial metabolites alter DNMT activity, the pattern of methyl marks changes. These shifts can silence or activate genes that control cell identity. In epithelial cells, reduced methylation at barrier gene promoters boosts their expression. Conversely, hypermethylation in immune cell loci dampens cytokine production. The net effect is a reprogramming of transcriptional networks that govern inflammation. Heat maps reveal clusters where methylation differences align with altered gene sets. Schematic diagrams show how demethylated enhancers recruit RNA polymerase Roman II more efficiently. Thus, microbial signals can indirectly shape host immunity through epigenetic rewiring. These changes are reversible if the metabolite environment is restored. Pause 0.8's microbial metabolites influence DNA methylation by competing with SAM, binding allosteric sites, or altering methyl donor availability. These interactions shift DNMT activity, reshaping epigenetic landscapes that govern host gene regulation.